In our last video, I left you with the question of which of the following transport mechanisms requires energy, and hopefully you answered that active transport requires energy. Only active transport requires energy. Simple diffusion, facilitated diffusion, and osmosis are all passive processes. Now, let's talk specifically about how plant cells use active transport of hydrogen to drive a bunch of different transport processes in their cells. So here we see what we call a proton pump. So this is a proton embedded in the membrane, the phospholipid bilayer here. And this proton pump, when an ATP donates a phosphate, and that phosphate attaches to the proton pump, that provides energy and changes the shape of the molecule such that hydrogen ions are pushed to the opposite side of the membrane. And notice that these hydrogen ions are moving against their concentration gradient from low hydrogens to high hydrogens. So this is an active transport. And by this time, you should be fairly familiar with a cell membrane having a higher concentration of hydrogens on one side than the other. And this is called a membrane potential. There's a potential difference in charge, and this difference in charge, and this we have called it an electrochemical gradient, and it is also sometimes known as a membrane potential. And it represents stored energy, this higher concentration of hydrogens on this side of the membrane compared to this side of the membrane. So the cell can take advantage of this stored energy in a couple of different ways. So the first way is that the hydrogen gradient can power co-transport. Co-transport is a type of transport that uses a transporter protein that has to bind to two different molecules in order for those molecules, or ions I should say, in order for those particles to be transported across the membrane. Co-transport can sometimes have molecules or particles moving in opposite directions, but in the cases that we're going to illustrate here, we will show the particles moving in the same direction as each other. And notice that the hydrogen ions are going to move down their concentration gradient from a high concentration to a low concentration, but the sucrose molecules are moving from where they have a low concentration to where they have a higher concentration. So the sucrose is being moved against its concentration gradient, and the energy that's providing that is the membrane potential, the difference in charge between the outside of the membrane and the inside of the membrane. So the outside is relatively positive, as you can see with these plus signs. The inside is relatively negative. And so that provides the potential energy to move the sucrose. And of course, when the sucrose is moving, that would be kinetic energy. So this is an example of using a neutral solute, and this is how cells load sucrose into their phloem tubes for moving sugar throughout the plant. But co-transport can also involve ions, and so in this example we see nitrate again moving against its concentration gradient from where nitrate is low to where nitrate is high. And again, it is the hydrogen ions that are moving down their concentration gradient that are providing the energy to allow the nitrate to move against its concentration gradient. And this is how roots absorb nitrates so that they can get nitrogen. And then why do plants need nitrogen? Well, it's because plants have to make their own amino acids. They don't eat protein, except for a few exceptions like carnivorous plants that eat insects, and then they get some of their nitrogen from the plants they eat, or from the animals they eat. But most plants rely on taking in nitrogen from their roots, and the form that they often use is nitrate, And this is made possible when the roots actually pump hydrogen into the soil so that they can then uptake the nitrate by this process of co-transport. And then another thing that this membrane potential does is that it often stimulates a channel to open in response to this membrane potential. So in this case, you can see a higher positive charge on this side of the membrane and a lower positive or a negative charge on the inside. And so potassium is going to move across this membrane from where it is in high concentration to where it is in low concentration. And this is allowed because this ion channel opens whenever this membrane potential happens. And so this can allow for facilitated diffusion of ions. The potassiums are going with their concentration gradient, but they wouldn't be able to be facilitated by this channel until there is a membrane potential that stimulates the opening of this protein. In case you're wondering how that maybe would happen, remember that if you have a bunch of hydrogen ions out here, they might actually change the hydrogen bonding within the protein, and that might actually change the shape of this protein slightly and make it so that potassium can start flowing through. All right, so to illustrate these concepts, I want to show you a video, and this video comes from the McGraw-Hill. It's kind of an older McGraw-Hill page, and this requires Flash, which I've enabled for this page, and I just think that it might be useful for us. So I'm going to unplug my headset, so hopefully you'll be able to.
Most of water and minerals absorbed by plants come in through root hairs, which collectively have an enormous surface area. Solutes cross the root hair membrane very slowly unless there are transport proteins in the membrane. Some of these proteins are selective and facilitate diffusion of specific minerals by binding the solute on one side of the membrane and releasing it on the other side. For example, potassium transport proteins allow potassium ions, but not sodium ions, to pass through. In addition to the passive transport proteins, there are proteins that utilize energy to transport ions across the cell membrane against a concentration gradient. One type of active transport mechanism in plants involves the use of the proton pump, where the energy released from ATP hydrolysis is used to pump hydrogen ions inside to the outside of the cell. This results in the proton concentration being higher on the outside of the cell than on the inside, and creates a proton gradient that represents a form of stored energy called the membrane potential. Plant cells use the energy of the membrane potential to drive the transport of different solutes into the cell. Because cations such as calcium ions are positively charged and the inside of the cell negatively charged, the membrane potential helps to drive cations into the cell through specific transport proteins. Cations move down the electrochemical gradient, driven by the active transport of hydrogen ions out of the cell. Negatively charged ions, such as nitrate, enter cells by a co-transport mechanism. Proteins that allow protons to re-enter the cell couple the transport of the protons to the movement of nitrate into the cell.
Hopefully that video was somewhat helpful for your understanding. So that leads us up to our next question, which has to do with the generation of an electrochemical proton gradient across membranes.